5, 6, only 6 of us here so far. Yeah. I mean, I get it.
Fire truck.
This world, they blocked off like all the very back row of.
Yeah.
So
It's
we're-
for the firefighting program, because
Yeah.
we're doing some kind of training this week.
I no one knew about that. I feel like.
There was an email.
No.
There was an email.
For most people.
Yeah, most people did not read the email. I will say that. Most people did not read the email. There was an email. I remember they sent it a couple weeks ago. It wasn't like recent.
Yeah.
That's what it was.
Yeah.
So yeah, so people like saw the email and they're like, whatever, there's something happening in the fire in the
Yeah.
parking lot. But then 2 weeks later, now we're having this, people are not connecting those yet.
Yeah.
Yeah, because I knew about the email. I had seen it, knew the dates, but it wasn't until I got in on Monday that I'm like, oh, yeah, right, because I saw it
Yeah.
all locked up.
Yeah.
Thankfully, it should only last this week.
Oh, the whole week.
Yeah.
Well, you can go back to the email, I guess.
Yeah. It was like the 20th to the 28th. It was like for an entire week, it was blocked off.
Thursday.
Yeah.
Yeah, I got the parking lot for my writing class in the morning. I thought, are we not in class? Then why is the whole thing?
Yeah.
Most of the classes is like, she said something, and then she got the class, why is it?
Yeah. It's unfortunate, but it's only a week, so no better.
Well, hang on, I don't want to take too long because we do need to jump into chapter 6 and start talking about genetics and everything like that. Chapter 6 is definitely the heavier of our 2 chapters. If you were making your concept maps, you probably noticed that as well. That chapter 8 is more of just like techniques that relate to chapter 6. And so we're going to spend most of our time in lecture today talking about chapter 6, talking about genetics. So replication, transcription, and translation, and then talking about bacterial operons as well. Bacterial operons, I know they feel like a lot, they feel like something weird, and most of us are already like, we're probably making our concept maps, even we're like, whatever that means, whatever an operon is. But it's us dipping our tiny little toe into genetic regulation, which is, trust me when I tell you, dipping our tiny little toe into it as compared to how eukaryotes regulate genetic expression, which is much more complicated. I say, luckily, you're taking microbiology, and so we have to focus on the bacteria, which are simpler. But at the same time, you guys eventually, if you haven't already taken physiology, you'll take physiology, and you might talk a little bit about that stuff there too. All right, so before we get into that, we have some visitors from the SSOC. We're going to talk about some of the resources. I was hoping that more people would have shown up by now. It's a small class anyway, but still it's about half of the small class. But I'll go ahead and let you guys give your spiel and give me the floor.
All right. Yeah, thank you, Dr. Yost, for having us. As she was saying, my name is Camille. I'm an academic success coach at the Student Success and Opportunity Center, and this is?
I'm Sydney. I'm the STEM lab coordinator.
Yeah, so our office helps you guys by providing services that can help you guys all succeed academically throughout the school year. We're located on the 2nd floor of the Learning Resource Center, which if you exit out through this door is literally right across the street or walkway, I guess. So we offer different services. As I said, academic success coaching. We have a writing center, a free computer lab that includes free printing services, and most importantly for your class, the STEM lab. And Sydney will take over from here.
Yeah, so like I said, my name is Sydney. I am one of the 2 tutors that handles the STEM lab. Basically, we will tutor you in anything related to STEM. So that would be this class, your math classes, physics, all of that fun stuff. A little bit of background about myself. I got my bachelor's in biochemistry and molecular biology, and I'm getting my master's in regenerative medicine. Yeah, fun stuff, right? But yeah, so we are open Monday through Friday. Monday through Thursday, it's 9 to 7:30, and then Friday it's 8:30 to 5. 5 is a hard deadline because that is when the building closes. Now, we are walk-in. You don't need an appointment, so just come on in. But being walk-ins, that means we are first come, first serve. So midterms, it gets really busy. There might be 10 people in there. We will try to get to you as fast as possible, but you will have to wait. So please don't actually wait until you really need help. Come in early. But yeah, any questions for me?
Now that for my part. Right, so as I said earlier, I'm an academic success coach, which essentially means my job is to help you guys develop the tools and skills to combat any academic challenge you might face this semester. Or two, I wanted to highlight today, okay, are time management and study skills. Time management is just us learning how to balance and allocate your time among all the competing priorities in your life, especially at OSU OKC, where we know many of you guys are full-time workers, caregivers to either children or other people in your life. So, but we want to make sure we're balancing that stuff along with school. And I should know I'm in a similar boat. I'm a full-time worker here and I go to grad school. So yeah, I know what the 5 to 7 p.m. classes are like. So we want to make what I can do as an academic success coach is help you guys develop tools that can help you keep track of all the things going on in your life and making sure you're blocking off time for it, especially when it comes to studying, especially all the complex material you're learning in microbiology right now. Time management is really important when it comes to studying because that is an essential part of developing effective study habits. Effective study habits is basically a balancing act between memorizing and recalling, retaining all the information and reviewing and recalling all of it. So for example, maybe you ended up cramming either the day before or a couple days before for the test. So while you had time to memorize and retain the information, that doesn't mean it gave your brain enough time to process it and thus recall it when you need it. So even though you might have burned your eyeballs out studying chapter 6, that doesn't mean once you get to the chapter 6 test, you suddenly can't recall that information, which is crucial in a subject like microbiology, where just a couple things off suddenly means you are looking for a different answer than what you initially thought it was. Along with managing your time effectively while studying, an important part of effective study habits is using methods that actually work for you. So while all of you guys are in the same class, some of you guys are sitting in the same row, you are not the same person. And so all of you are different learners in different ways. So some of you guys might be visual learners, some of you guys might be kinesthetic learners who learn by doing, such as Kahoots. Some of you guys are okay with reading and writing, and some of you guys are auditory learners. So my job as an academic success coach is to help you figure out your learning style and help you select the best method that will actually help you understand the information you're learning. Okay? And you can do this by either booking a session with me, myself, Antonio, or Evelyn, who are the other academic success coaches across the walkway. Okay, we accept walk-ins similar to the STEM lab, but we also take booking appointments as well. Also, you can actually find time on your schedule to book an hour-long session with us. Do you have any questions about anything I've talked about or the academic success coaching in general? Okay. Thank you, Dr. Yost, for having us.
Yeah, thanks for coming out. Definitely make use of the resources at the SSOC if you haven't been to the STEM lab or the SSOC in general. It's a great study location. It's a nicer building than this one. And they have lots of open space, study carrels, and lots of people up there that can help you with your studying as well. If you haven't been over there, get the LRC, which is that big building just right across the walkway here.
Yes.
The SSOC is on the second floor, and then, the library and everything's over there as well. It's a great area for studying, lots of resources, and you're already paying for it. You're paying for it as part of your tuition, so you might as well make use of it. When I talked to you guys after the Unit 1 exam about, hey, now we know what's expected on the test, and if you want to do a little better, and one of my biggest recommendations is to talk about it or teach it to somebody else. And Sydney's a great person to talk that stuff through with because she's had to learn this stuff before, a lot of the stuff that she's familiar with. Now you have somebody you can go to, doesn't have to have your study group, bring your whole study group there and have them talk to the stuff. So it doesn't just have to be me. You have other people that are knowledgeable about the subject too that can help you with that stuff. And even if there's a topic that maybe she's not familiar with, she has the background to help you figure it out and to work with you guys on that. So, and if we get to that like more complicated stuff and start talking about how antibiotics work or the different ways to control microbial growth, or when we get into immunology, that would be stuff we might have a little bit more support with, and stuff especially. They'll be a great resource for that. I appreciate you guys coming in. If you guys want, they have their, rack sheets, and, cards and little pens and goodies and stuff up here. If you guys want to come up here and grab any of that, now would be a good time for that.
And something else I actually brought to your last class was, because we wanted to talk about time management and study skills, something we brought out to was a study plan that we actually give out at the SSOC, where it lets you plan out your week for studying, but also what methods and preparations you can do. And we actually have a list of that on there as well. And we have all of the academic success coaches business cards, including the STEM lab and pens as gifts for you guys. Okay, but thank you so much.
Yeah, thank you guys for coming out. I appreciate it.
Of course. And we're gonna go get our stuff too. But
Yeah. Absolutely. Do you guys have a key to open up that door? You need,
No, we do not.
I think it's locked.
Yeah.
If you want, I could also, if you wanna just keep it in for your late
I'll
class,
keep
I
it.
can
I-
get it tomorrow.
And I can always just bring it by. I'll have time during my office hours. I can bring it
Yeah.
by.
Yeah, I can also
Yeah.
just get it tomorrow. Thank you.
Yeah. Thank you, guys. Alrighty, so make sure that we're signed in, double check that we are signed in, and we're gonna get going with the Kahoot next. Who we got on here? I think it's gonna be Cassie. Okay. Okay. Alrighty, let's get going with chapter 6 here. Thing died, so I can't. There we go, okay. Let's just start and then hopefully make our way through it. All right, our first question. What is the term that refers to an organism's total sum of genetic material? So Azalee, you'll be my first victim after we answer this question, FYI. Just so you know, it'll be about one of the other terms up here. Okay, so the correct term for the total sum of the genetic information is going to be genome. So chromosome is one of our wrong answers. Do you know what a chromosome is? Roughly. Best guess here, best guess. So, okay, so I'll settle for that, and here's why. Because a chromosome really is just a structural unit of your DNA. And that really is. It doesn't have any functional aspect to itself other than it is a structural unit. So I'll settle for that because, I mean, how else do you define it other than it's part of the DNA? And it is just part. And that's another key aspect of it too. It's just part. So all the chromosomes that you have in your body, you have 23 pairs of chromosomes as a human being. Bacteria have one chromosome, not a pair, not pairs, just one chromosome. And it's in a big old circle. We have 23 pairs. Is that everything? Is that all for those organisms? No, right? So we even talked about our mitochondria. Your mitochondria have their own DNA. It's not encoded in your chromosomes. So that's extra chromosomal DNA is what we would call that. So extra chromosomal means outside of the chromosome. Bacteria also have extra chromosomal DNA. So they don't have a mitochondria. Obviously, those are membrane-bound organelles. What they do have are plasmids. So that's the one in red there. The plasmid is an extra bit of DNA. It's in a circle, has extra genes that can help with survival, but aren't essential for survival. So what it means is if you take out the plasmid, bacteria can still survive. Maybe not as well, but it won't die. But if we take out the chromosome, they die, right? They can't actually live anymore. So that's the idea with the plasmid. They're extra bits of DNA. What kind of DNA might be on a plasmid? That might be genes like antibiotic resistance or certain toxins, things like that. Yeah. So good job. Let's move on. We're going to spend most of our time on chapter 6. I'm going to try to cover it as much as I can. So let's get cranking with it. All right. The physical expression of a genotype. What's the term for that one? There won't be any questions about this one. Only so much I can get at it. Physical expression of a genotype. So this is phenotype. And that is how I remember it too, okay? Your genotype is your genes, what your genes are. Your genes don't mean anything if they're not actually making the product. The actual genes in your cells, that's your genotype, all right? What's expressed by your genes, having red hair or brown hair, blue eyes or green eyes, or being a red, pink, or white flower, for example, that physical expression is the phenotype. Genes, genotype, physical phenotype, okay? All right, so that's how they're related. We can see here, we've got big R and little r, and those are alleles. Okay, so there's different versions of that gene that you can have. Here, we're just talking about two different versions, big R version and little r version. Big R means that it is a dominant trait, and little r means that it is recessive. And our chapter doesn't go about that in a whole lot of detail. You're not going to have to learn a ton about genetics and Punnett squares and things like that in this course, okay? Not in this course. But it is worth knowing a little bit about genetics and how genes are expressed. So with the dominant and the recessive traits here, the big R means that it's dominant. That means that it's generally going to be expressed preferably over the little r trait. Here we have a mixing of the two traits. Okay, so that's a unique sort of a situation there. Anyways, let's move on. The 3 parts to a DNA nucleotide. Here's where we're getting into the meat of things. All right, so the 3 parts to a DNA nucleotide. Deoxyribose sugar. Now, if it was RNA, it would be ribose sugar. All right. Phosphate and a nitrogenous base. Nitrogenous base is a fancy word, okay? But basically, the way DNA, we think of DNA as being kind of like a ladder. We've got the sides of the ladder and the rungs of the ladder. And we typically see it drawn as a helix. Right? And you can imagine untwisting that helix to get that flat ladder, right? So this is our general structure of DNA. But what's actually on the backbone here is alternating sugar phosphates, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. The other side of the ladder, I'm gonna draw it a little bit more over here, give me some space, all right, is a phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. All right. Attached to these sugars is that nitrogenous base. So, who do we have next? Kaylee's not here. I think she said she might be late, or she might not be here today, actually. All right, Dylan is our next person. Dylan, what are our 4 DNA bases? You can just do the letters if you want to. Good, excellent. Okay, so for DNA, those nitrogenous bases, the B here, I put a B here just to represent any of those 4, all right? But those bases, the B, let me give myself a little space here, are attached to the sugar. And then DNA bases are A, T, C, or G. And what does A bind to? C. And what's C bind to? Obviously, G. Okay. In DNA, those are our bases. All right. So imagine that the B, if this B was a T, that other one would be an A, and whatever. I'm not going to draw out the whole thing then. There is a button on the side of this pen, and I keep pressing it when I'm trying to draw. So please excuse me while I accommodate that. Oh my gosh. This. Okay. So we get the gist of it, right? And then what type of, let me actually ask the person a question. We're going to get almost all of our questions out of the way pretty early on because I want to be sure that our basics for the genetic stuff is taken care of. All right, so Duncan is our next person. These bases, when we talk about A binding with T or C binding with G, what type of bond holds them together? So we're creating that double helix because these guys are bonded together. So if we had to choose between 3 types of chemical bonds that we talked about, covalent, ionic, or hydrogen bonds, Hydrogen bond. There, that's exactly what we're getting. I knew you knew it. I just needed to get you to that. All right. So A, when we say A binds with T, we mean that A and T develop hydrogen bonds with one another. Now, it's like 3 hydrogen bonds or it's 2 hydrogen bonds, depending on what bases we're talking about, and whether you're a pyrimidine or a purine. I'm not going to test you on purines and pyrimidines. You're welcome. I'm also not going to test you on the structures or which ones have 3 bonds versus 2 bonds. I don't care as much about that. I want to get the foundation of how the genetics work down, okay? So we're going to be focusing mostly on the processes of replication, transcription, and translation, all right? So I'm drawing all this because it relates to replication, transcription, and translation. All right, so yes, hydrogen bonds, that's the type of bond that holds our bases together. And this is going to be a question coming up in the Kahoot. And yes, it's going to be a question that's probably going to be on the test. All right, so to show this a little bit more clearly, the question says the 3 parts to a DNA nucleotide. We think of a nucleotide as our building block for DNA, right? What is a nucleotide actually? It's this. This is a nucleotide. This is a nucleotide. This is a nucleotide. And this is a nucleotide. On the other side, this is a nucleotide. Now, you notice how these guys are oriented sort of the opposite direction of one another? If we were to look at the actual chemical structural makeup of DNA, it's the sugar that sort of dictates this, right? It's a 5-carbon sugar. And basically, whether that sugar is facing up like this or facing down, just bear with me with my drawings, like that, determines how that backbone is facing, all right? And we number those sugars based on what they're attached to. You guys remember that from chemistry, right? Numbering those carbons, the carbons in the sugar based on what those are attached to. And I'm not gonna, like I said, I'm not gonna give you a picture of a sugar and make you number the carbons or anything like that. I'm not gonna show you a nucleotide and be like, which one's the 5, which one's the 3? I don't care, okay? I don't care. But when you start talking about DNA, you do need to have some idea of what it means when we say it's oriented in a 5' to 3' direction, which is also one of those questions coming up. So what that means is sugar carbon number 5 is at one end, which is always what we consider our start end, okay? This is the 5 end. And so what's at the other end? The 3, right? So the other one, this would be the 5 end, and this would be the 3. So we notice that one of them is running in one direction and one of them is running in the other direction. And there is a term for that. Jonathan, do you know what the term is?
You look like during replication.
Not necessarily during replication, but the fact that those 2 strands, because DNA is double stranded. So each of those strands, the backbones are running in opposite direction, and that makes this, that is it, exactly, anti-parallel. That is what anti-parallel means, that our backbones are running in opposite directions. Beautiful. First person to actually just recall the term and not have to look it up online. All right. That's why I like this class better. I won't tell anybody else who said that. All right. Okay. Okay. Let me see. We don't, Sonia's not here, right? So Sonia's not here. She usually sits by you, right? Okay. Thought so. I just want to be sure. She would be our next person, but she's not here yet. Anyways. I'm trying to think, is there anything else I want to ask yet? So this is the idea of how DNA is set up. We've got those 2 anti-parallel backbones. We've got the bases that are hydrogen bonded in the middle. I think I'm going to leave that there, and we'll get into replication probably in the questions here in a second, and then I'll pick up. All right. DNA anti-parallel strands of nucleotides. How are the strands held together? How are the A's and T's or the G's and C's held together? Dylan answered this for us, so we should be able to recall it. Not Dylan, Duncan, sorry. I don't know why, sorry. I was just looking at my list over here. All right. I was gonna write on it, I don't need it yet. Okay. Yes, beautiful, perfect. All righty, beautiful. So, good. I don't know what else to say about this. We already talked about it, so we're moving on. True or false? DNA polymerase builds DNA in the 5' to 3' direction. So we know how the strands are oriented. Which direction does DNA polymerase add on the nucleotides? From what end to what end? Yes, so this is true. It works in the 5 to 3. I know it's opposite from the numbers and what order they go in. So you're just going to have to just repeat that in your mind that it's always 5 to 3. Always. Okay. So this is going to get us into DNA replication. So let's get some basics down first. When we are replicating DNA, first of all, DNA replication, we're going to learn about 3 processes, right? I'll write them down here at the bottom. Replication, transcription, and translation. Replication is making copies of DNA. You're going from DNA to DNA. You're using DNA as a template to make more DNA. You're going from 1 strand to 2 strands in each cycle of replication. And that might sound familiar because that's exactly how PCR works as well. You go from 1 strand to 2 strands, okay? And that would be considered an exponential process. All right. Next person, Jillian. What is transcription?
It's changing from DNA to mRNA.
Exactly. So DNA to RNA is transcription. And then our next person is Tanya, who is not here either. Again, yet. All right. Evelyn. What is translation?
To amino acids.
Beautiful. RNA to amino acids, which build proteins, right? Okay, so that's our general processes. These are our big picture terms. I'm going to leave them there. And yeah, we're probably going to have questions about them. We're going to leave them there so we can keep in mind what all of these things are and how they're related. So anytime that you are needing to make a copy of your DNA for some reason, you're making a new cell, you'll have to copy your DNA, like we talked about with mitosis, right? In mitosis, we took a chromosome, and during DNA replication, we took 1 chromosome and turned it into 2, linked at the center, and then we pulled them apart. So we had to go through replication, and then while they were linked at the center, then pulled them apart so that each cell now had a copy of each chromosome. And that was mitosis. That's what happened in mitosis. So we learned about that in chapter 5 a little bit, right? Very briefly. Okay. So when you were an egg getting fertilized by a sperm, you had 23 chromosomes, only 23, not 23 pairs in the egg, just 23 single chromosomes in one egg. And then the sperm had 23 chromosomes and they came together and now you had 23 pairs after they came together. So that was a full cell at that point, right? And you were just 1 cell at that point after fertilization. So in order to go from 1 cell to the millions that you are now, we had to copy our DNA. We went through DNA replication and then went through mitosis in order to build those cells and turn you into a baby and then a child and then an adult. And then when you cut your hand, when you're cooking to repair that, we had to make new cells. We had to replicate that as well. So I'm thinking about this over time. Now you're like, yes, I know how replication works. And I'm proud of you, okay? But I also want to mention something, which is while we were doing all that, you see that I always want to ask somebody, can you just take off your shoelace so I can demonstrate this? But this is that's a weird question, so I'm not going to do that. Let me try to take one of these, see if I can show it. Take a flat, long thing. Imagine that this is very, very long. And we think of DNA as being a helix, and it's turned like that, and it's all helical and happy. But imagine you're taking a helix that's twisted like that, and you just keep twisting it, and keep twisting it, and keep twisting it. We did that, and I can't do it with a tiny piece of paper, so it's maybe not a good example. But if I took a shoelace that's flat like that also, and twisted it until it was in like a helix, almost like a tube, eventually it would turn up to. But keep twisting it. Then that's going to start twisting on itself. And then that's going to start twisting on itself, and so on and so forth. That's how DNA is actually packaged in your body. Multi-twists on multi-twists. We twist it up and then twist it further until it twists on itself, but it's a regulated twist. All of those twists are managed by other proteins called histones, which we're not going to get into what each of the histones does, but they basically manage the twists on how the helix is going to twist back on itself. So we can package it nice and neat in your nucleus. And that's how we keep all the DNA organized inside of your nucleus. It's wrapped around these histones. But now we need to make copies of it. Or we need to access a gene and transcribe just that gene. So now you have to flatten part of it open and open it up and access that gene. Or if you're going to make copies of it, flatten part of it open and open it up and gradually open and open and open and open and open and open until we get to the ends. But it's the same as whatever we were talking about with your shoelace. If it was all wound up, super tight helix, and you open it at parts and start spreading it apart, what happens to the ends? If you were to hold on to the ends and keep them stationary, what would happen? We're going to consolidate a lot of twisting and tension at the ends. They can't unwind. Those are your telomeres. That's where all that tension is held in your chromosomes. Every time that we copy a chromosome, you lose a little bit of telomere because we can't unwind it completely to make a copy of it. It's just physics, quite literally. And so when we talk about how whenever you get older, your telomeres shorten, that's why. Because from the moment you are a fertilized egg and you have to replicate those bits of chromosome, you start shortening them little by little by little. And the telomeres are just extra bits of DNA that are meant to accommodate that. So that's nice. But at some point, if you get old enough and you live long enough, you're not going to have any more telomere left. And you're going to start losing genetic information because physics said you can't get rid of it. You can't, that process doesn't stop just because we started hitting genetic information. So that can lead to problems over time. It's also a problem with cloning. If I clone a cell from an adult that has already lost telomere, then that new baby that we made from that adult starts with a shortened telomere. This is a problem with Dolly the sheep as well, just so we know. All right, so that's an issue with cloning. If you want to clone somebody, you want to clone them from a very, very young age so you don't lose telomeres there. How big of an issue is this? How much does it affect us? And things like that. That's a bigger question that we're not going to get into. Okay. Can you prevent your telomeres from shortening? No, absolutely not. Not yet. Right now, we don't have any way to lengthen your telomeres and gain you back more telomere cushion, basically. Okay. So anyways, you want to look into that? It is worth knowing, but that is a process that also happens with our DNA replication. So in DNA, we are going from basically one double-stranded DNA bear with me while I do the colors because I promise you'll thank me for it later, okay? To two double-stranded DNA. All right, and this is, see how we started with two, like pink, and we went to a pink and a blue. I'm going to show you how that process works and what that means and everything as well as we walk through this. So basically, you'll start off with this, and of course, this is going on and on forever and ever. In both directions, all right? So if this is the 5 at this end, 3 is up here, but we also know that the 3 is at this end and the 5 is down here. Just to bring it back to this drawing over here, it's still the same situation. I'm just not drawing out the S's and the P's and the B's. Yeah, okay. So we got our little ladder and then we open it up here. Anybody remember what this structure is called in replication? The replication fork. And the videos that I have on the concept map thing do this better justice than my drawings do, but I don't know, for what it's worth, I'm going to try to explain it through, okay? All right. If we can only add nucleotides in the 5' to 3' direction, and this new strand, let's do blue. Well, let me start. First of all, DNA polymerase. We're going to talk about DNA polymerase. We're going to talk about another enzyme, DNA ligase. We talked about helicase. Helicase is an enzyme that unwinds the DNA, okay? So there's some enzymes there. You're noticing a trend with these enzymes, how their names all end in ACE? Almost always, if you hear something ACE, it's a protein that functions as an enzyme. And an enzyme is a catalyst for a chemical reaction. You guys remember catalysts from chemistry? The catalyst is something that helps overcome activation energy. It lowers the amount of energy you need to put into a reaction so that it can happen more easily. And it's usually not changed permanently in the reaction. That's going to be enzymes. Chapter 10 covers enzymes and metabolism. But just to lead into that, we're already talking about enzymes and their functions. So DNA polymerase, it does tell you what it is. ACE means it's an enzyme. DNA polymer means that we're going to make DNA polymers, which means we're going to take nucleotides and build DNA. It's DNA polymerase. That's what it is. DNA ligase, it tells you what it does too. What it's telling you is that it's going to ligate DNA. Ligate means to join together. I used to play clarinet when I and saxophone. And I had a ligature. The ligature joins the reed to the mouthpiece. That's what ligate means, to join, okay? All right, so the problem is that DNA polymerase can't just add on nucleotides to nothing. It has to add on nucleotides to something else. And so we have to start with a primer. We're going to prime first. What's this primer made of? Does anybody know? It's RNA. We'll come and replace it later. It'll be several nucleotides long, FYI, but I'm putting it as just a dot for now, okay? Anyways, DNA polymerase from the 5' end will add on nucleotides in the 5' to 3' direction. This is easy enough for it. Now, we will be building one of these two strands. You see how it's going to be one pink and one blue? One part old, one part new. So DNA replication is a process because we have one part old and one part new. Jessica, what's the term for that? We say that DNA replication is?
Oh. I don't know. That's okay.
Yes. You can take a minute if you need to. You want me to give you a minute?
Yeah.
Okay, so we'll come back to it. So we'll be making two new strands of DNA, each of them consisting one part old and one part newly synthesized. All right, so that's the idea of how this will work. The other strand, actually, I'll ask you another question. So see if you know this one.
Okay.
So this strand that I just drew here is made continuously. We can just feeding in to that replication fork and just keep feeding in as we're opening that fork, opening up and feeding in. So what's the term for that continuously synthesized strand?
A leading strand.
Exactly. So we'll give you that one. That's a little, and you'll know what this next term is. It's just maybe I wasn't wording it in a way to make it obvious. So because our new strands here, our strand 1 and strand 2, where I have the stars written up here, are part old and part new. Let's see if Maria got it. Semi-conservative. That's yes. So you know this term, right? It's semi-conservative. Each new strand is part old and part new. We have with semi-conservative because we've semi-conserved the old strand. Make sense? Okay. So that's where semi-conservative is. Beautiful. We have old and new. Beautiful. Okay. So we're tracking a little bit, right? This other strand, can we just also feed it in like that? Can we just go straight in? No. Okay, beautiful. So we're getting that. All right. So we can't just feed straight in like that. So what we can do, I need another, yes, we need the primers. We can make it in parts. We'll add a little primer. And then we will feed off of that primer. Because we have to add in the 5' to 3'. We always have to be building in 5' to 3'. Yes, that one has a primer too. Don't worry, I didn't forget. Okay. So this strand on the inside, because we always have to be building in the 5' to 3', as we open up the fork, open up some more nucleotides that are free, and then come in and fill in that spot. This is synthesized in fragments. And anybody remember what those fragments are called? Okazaki. Beautiful. Be able to recognize it on the test, right? Might not be able to say it. I'm not going to make you speak the word necessarily, except for in class here, but we won't judge you. These are Okazaki fragments. All right. And when Julie, what do we call that strand? So not leading, but I'm betting that Maria has the answer here. Do you remember? I'll give you a minute. I'll let you cook with that one, okay? Yes. All right, so lagging, there it is. So the leading strand is made continuously. The lagging strand, I don't know why I wrote it there, but I wish this thing had, the only thing I wish this had is an undo button. The lagging strand is Okazaki fragments. Oh, okay, there's a check mark there now. Apparently that's a thing. Beautiful. I'm assuming that's what we put there too. That's beautiful. Okay. All right, so we're tracking this, right? Okay, so now we have DNA that's synthesized, but we got these fragments on our lagging strand, and that's a problem because later, whenever we have the whole DNA made and we separate them into separate cells and everything like that, we can't have that new DNA partially in fragments. So we need to join those fragments together. This is where that enzyme that I mentioned previously, DNA ligase, comes in. DNA ligase comes in and seals the fragments together. And DNA ligase always has that function, whether we're using it here or when we're using it in our genetic engineering chapter, when we're sealing fragments potentially there as well, okay? So that's DNA ligase. I think that pretty much sums up replication. So let's see how far we can get into our replication questions here. I'm going to leave it up. You can use it as a cheat sheet if you want to for the next questions. Which enzyme connects our Okazaki fragments formed during DNA replication? Don't worry about it. I like to have some kind of picture, and sometimes it doesn't suggest good ones. Alrighty, so that's DNA ligase. That's our enzyme that joins those fragments. I'm freeing up some space because we all know what's coming next, and that's going to be transcription. We already know that transcription is going to be making RNA from our DNA source. So I'm going to leave our replication thing up here because it's good reference for our structure of our DNA. So good. All right. Next question, during transcription, what happens? And it is up on here. Transcription, what are we doing? We've already gotten through everybody here. Yeah, everybody's been asked a question. So now it's just, I'm assuming that you guys will volunteer participation. Okay. So RNA is produced during transcription. Okay, so here is the idea with this. You start with DNA. I can't actually click on it with this pen. It's annoying me. Okay. Let's see. Let's start with our DNA is still pink. We start with DNA. We're going to separate our DNA. And yes, it'll be open in a replication fork, just like our picture here, but this is a better picture, right? Can we agree? Okay, so there's that. All right, so DNA. If this side of the DNA was A, T, C, G, I don't know, G, who cares? Then the other side would have been T, T, A, G, C, C, right? Okay. We're tracking that at least. One of these strands will serve as a coding strand and the other one as a template. What does this mean? Well, what it means as the template, when we come in to make RNA, which I'm going to draw as, who cares what color? Blue, sure, ice blue, sounds great. RNA will come in and match. Let's say if we're using the template strand, RNA will match up to the template strand. But its code should be the same as the coding strand, essentially, but replacing T with U. Beautiful. So this would still read A, but what would go here? U. Beautiful. Okay, you got that? Makes sense? So we're still opening up, just like we had a replication fork, except it's not a replication fork because we're not going through replication. We're going through transcription. And instead of DNA polymerase, what enzyme is going to be driving transcription? RNA polymerase. Okay. RNA polymerase always starts transcription at the same spot, and that spot is called the promoter region. So we'll start, E is the promoter region. Promoter. This will be more important when we get into the operons later. Okay. All right, so we'll make our RNA. Whether you are making ribosomal RNA, tRNA, or messenger RNA, transcription is the same process. It's just with ribosomal RNA and tRNA, this is your end product. With the mRNA, it's our middleman, right? Because remember, DNA is going to be our blueprint. We're going to make a copy of the gene that is our working copy, that's our RNA. I'm going to break it down even more, so don't worry. Then we're going to use that working copy and make our protein. So let's break it down even more. Imagine you have a huge, massive book of recipes of food that represents all of the food that you could ever eat in your life. Any recipe for any food that you could eat in your life, right? And you have to cook any food from scratch that you're going to eat in your life. This is a big cookbook, okay? And it sits in your pantry, but it's so big, that sucker ain't coming out of your pantry, okay? So that cookbook, the compendium, anything you could ever want to build, ever, recipe-wise, cook recipe-wise, okay? Stays in your pantry because it's so big and you couldn't move it if you wanted to. It's too heavy anyways, okay? But it's in your pantry. So tonight you want to make spaghetti and meatballs, which are my doggy's names, okay? You want to make spaghetti and meatballs tonight. So you're going to go to that big old book. You're going to open it to spaghetti and meatballs, and you're going to take a sheet of paper and you're going to write down the recipe for spaghetti and meatballs, just that one, all right? And you're going to take it out to the kitchen where you're going to cook the spaghetti and meatballs. That process, you guys know what a scribe is. Yes, it's somebody that writes something down. You were a scribe transcribing the typed out recipe into a handwritten version of just the spaghettis and meatballs. So it's still the same language too. We went from DNA, which is a nucleic acid, to RNA, which is a nucleic acid. But think of DNA as that big typed fancy cookbook version. And now RNA is your little handwritten temporary version. So we transcribed it. We just wrote it. Okay. All right. Now we're going to take it into the kitchen and cook our meal. Now, this isn't as easy and direct translations across these things, but if we have our RNA, our working copy of our gene, our copy of our spaghetti and meatballs recipe, obviously that recipe isn't the spaghettis and meatballs that we want to eat. So now we have to follow the recipe and build it. Well, it calls for flour, it calls for eggs, it calls for this. And each step of the recipe calls for a different thing. Each step of the recipe you can think of as the codons of our RNA. Those are the 3 nucleotides of an RNA. So AUG is a codon coding for the amino acid methionine. Think of that as this step called for eggs. So methionine is the eggs, for example, okay? All right. So we haven't quite gotten into translation yet, but that's the idea. And you notice how I skipped over transcription? Because transcription as a process is very similar to replication. It's just instead of making DNA, you're making RNA. RNA tends to be single-stranded. It's also very temporary in our body. It's a working copy. Once you've made your spaghetti and meatballs, the one that you wrote down, you're just going to throw it away. Because you know, back in the DNA, you can always get another, you can always come back in there and transcribe a new copy of it. So it's temporary while we're working with it. Then once we're done, our body disposes of it. It's the same thing that happens when you get a COVID vaccine. When you get a COVID vaccine, we are injecting you with mRNA. Your body will translate it into the product, which is the spike protein for the virus in your arm or whatever. And so your immune system can react to it. But once we've made that spike protein, your body gets done with that RNA. Did you notice how at no point did I say after making your RNA, you then go back and insert it into your DNA? It's literally impossible. This is impossible as me turning into a puppy or spaghetti and meatballs right now. That impossible. So your cells do not have the machinery to turn RNA back into DNA. Some viruses carry that machinery, like HIV. Okay, we're going to talk about it soon. All right. But this process, your cells are only built to go from DNA to RNA, and it cannot convert the RNA back into DNA, which is why it is absolutely false for anybody to believe that you can get an mRNA vaccine and it messes up your DNA. It's impossible. Are we clear and agreed on this? Please, yes. Okay. So, am I saying there are no dangers with mRNA vaccines? That is not what I'm saying. There are dangers associated with all vaccines, probably even mRNA vaccines. They're so new right now, we probably haven't been able to really figure out what the risks really are, okay? But this is not one of them. They don't become DNA and alter your DNA. Right now, as I talked to my mom this weekend, my back is hurting, so you gotta excuse me. I talked to my mom this last weekend, and she had some things to say. Oh, we're gonna be moving towards mRNA vaccines. You know, that's gonna be a problem because they change our DNA. So I explained this to her, okay? Now you guys are educated on it too. Then I said, you know, Mom, this is the situation, and I educated her on it. And she's like, oh, yeah, well, you know, it's also causing harm. There's always some reason, yes? It's also causing harm. It's causing heart damage. And I think we mentioned this briefly, but I wanted to go ahead and talk about this because I did my research, and I'm wondering if anybody else did. I did my due diligence. I looked up the research that may or may not support there being heart damage associated with the COVID vaccine. There is correlation, but not causation yet. This is hard to do because most Americans have been vaccinated with the COVID vaccine. So how do you show that the COVID vaccine is causing heart disease and not just aging or smoking or vaping, for crying out loud, or something like that, right? So that part, we haven't delineated the causation. Is it possible that the COVID vaccine might have caused a slight increase in what specifically is cardiac artery disease? So we're talking about heart attacks potentially resulting from this, okay? Possible, it is possible. So I'm not going to deny it, but it seems to be a very low incidence of increase. Now, shall we talk about what happens when you get COVID? There is a high incidence of heart disease associated with actually having COVID if you are unvaccinated. Do we not remember what COVID was like before we got vaccinated? I'm hoping we can reach back the 5 years ago that it was and remember how scared we were, because otherwise healthy people were hospitalized, dying on ventilators, and having heart disease developing from having had COVID. And years down the line, may continue to have heart disease from having had COVID previously. There's a high incidence of that risk associated with having had COVID. There's a small risk of that with the vaccine. Now you can choose not to get the vaccine and take your chances having COVID. And the impact, when I say this, won't be as big in this class. But how many of you guys have had COVID at any point? Raise your hands. Okay. Most of my other classes, we're talking 20, 25 people classes. I might have one, maybe, that didn't raise their hand. I also want to remind you guys, 1/3 of people that will have COVID will not have any symptoms at all. So those of you that may not have raised your hands may have had COVID. You just didn't know. Or because we have had the vaccines since then, if you didn't test and you got a little tiny cold and you didn't test during that time, you may have had COVID. And also, I'm one of those people, when I had COVID, I was sick. I'm talking 104.5-degree fever after I got vaccinated, okay? I was very sick. I almost had to go to the hospital because my fever was getting so high. I had to take ice baths because of it, all right? That was after I got vaccinated. I did not test positive the entire time I was sick until the day I started feeling better. And then I tested positive. Some people's bodies just react that way, how they're making the virus and where it's expressed and where it's presenting in their bodies. So you may have had COVID and not know also. You tested negative, it felt like crap, but how do you know it wasn't really COVID? So that's another thing too. So probably odds are probably 90 to 95% of us by now have had some version of COVID. Most of us after the vaccines were instilled. Some of us before. And if you had it before, you probably had a much more serious disease than those of us who got it after the vaccinations were introduced. So how do we even compare it? It's kind of hard to compare at that point. So now the issue is, should I get the third booster? Now I'm gonna be completely transparent with you guys. I haven't had my third shot. I got the first 2 that were required for you to go back to work and stuff like that, right? I got those 2. But I never got my third shot, even though they were promoting it. I just was lazy. That's my literal excuse, okay? I haven't done it because I was lazy, okay? As it turns out, if you get the third shot, your incidence for heart disease goes down. So if you are worried about the heart disease associated with the vaccine, get your third booster if you haven't yet, okay? Yes, so me, I haven't yet, but now I haven't learned that because I did my due diligence, I did my research. So I'm probably gonna get my third vaccine so I can reduce my risk for heart disease associated with the COVID vaccine. Why would that be? Well, we haven't learned immunology yet, that's unit 3, okay? So it might be a better discussion for unit 3 to have that. But the gist of it is that when you had the first 2 vaccines, your body was trained to see this antigen as a virus, not as a, oh my gosh, end all be all, we need to fire the missiles, fire the missiles, fire the missiles, which is what happens in COVID without the vaccine. Instead, it's like we need to send a tactical team to go fight this guy and take care of him quietly, like it should be. And so the third vaccine just enforces that even more. So the second one helps remind you need to call in the troops that are just specialized. Okay, guys? Yeah, sure, dad, whatever. And the third one says, no, for sure, only send in the specialists. We're not going to overreact to anything. We think that the inflammation involved with the overreaction is what is leading to the heart disease. We think, but we haven't proven it yet, but there's a lot of evidence that suggests that COVID antigens in general can incite an inflammatory response generally. So FYI, I hope we feel better educated now about that. Okay, so this is transcription. It works the same as the replication, but instead of making double-stranded DNA, we're making a single-stranded RNA. Yep, we see that in the picture. It's better than I can draw it. So hopefully we can see that. Okay, so that's transcription. Because we also know that after we make our RNA, we're going to go through translation. So now we have our RNA. Let me erase this because this is a mess. We know how we get the RNA. Just keep that in mind. We went from a double-stranded DNA. Erase all this. I want to keep that other stuff on the other side because I just feel like it ties in well. Now we're going to make our RNA. Okay. And with that RNA, we might have, I don't know, AUG. This is our RNA, mRNA. No, it doesn't want me to do it. I want to be very specific here. mRNA. AUG, we might have CCC, and who knows, who knows? Okay, great, sounds good, right? All right, so this big old structure comes and clamps on to that mRNA during translation. What's this structure? A ribosome. What are ribosomes made of? Easy enough. This is our RNA, ribosomal RNA. So it's a big old structure. It is made out of RNA also, and some proteins in there as well. It's a little more complicated than that, but largely made up of ribosomal RNA. But for our functional purpose, it's this blob, all right? Okay. So that happens. And each of these three, and you notice that I wrote them in threes, each of these three nucleotides, these bases, we call them a codon. In the DNA, that codon was known as a triplet. So the DNA GGG, and we made the RNA CCC. That DNA GGG was our triplet. And RNA, it's the codon. Okay, there's a difference there. All right, so something comes in and it binds to these. This looks like this. It's fancy, real fancy. And here is attached to an amino acid. In AUG, on our, you guys know what this structure is that I'm drawing? It looks like a guitar. It's not meant to, but it is a tRNA. All right. So the first one comes into AUG, and on the tRNA, it's RNA also, but we have a special part of the RNA that would read UAC. You see how it's the complement to it? You guys remember what this is called? If the blue is the codon, if the blue on the mRNA is the codon, the tRNA sequence that matches it is known as the anticodon. Beautiful. Again, the video is better at showing the whole process and how it continues to work than my drawing is, but my drawing allows me to break it down a little more. So because the mRNA says AUG, it has adenine, uracil, and guanine. Because it has those three in that order, it matches up with uracil, adenine, cytosine on our tRNA. And because those match up, this amino acid that was carried with it, methionine, is our first amino acid. Then our next one comes in, that is GGG, whatever, it's attached to something else. Here we go. I don't know all of my codons, so you'll have to, bear with me. Let's say it's lysine. Who knows? All right. Then we have UUU, and this matches up with AAA. And then this would code for, and we usually really only have two of them coming in at a time, and we'll just shift down and move to the next guy, shift down and move to the next guy, and whatever. Let's say that this codes for, I don't even know, isoleucine, ILE. All right. And we're gonna link each of these amino acids. This is amino acid, and that's an amino acid. So each of these is an amino acid. What kind of bond comes between amino acids? Once that peptide bond is formed, we can get rid of the tRNA. And so it'll exit through the E site. And we can move down our ribosome and keep adding on our amino acids. So each 3 bases on our mRNA codes for 1 amino acid. And there is what we call wobble. So GGG might code for glutamate, but GGA might also code for glutamate. Okay, so there's some what we call the wobble effect in that 3rd nucleotide position sometimes. As it turns out, AUG only and always codes for methionine. There are some other ones that are like that too, but very few of them have 1 match. All right, there are 20 amino acids. So based on those combinations of A, U, C, and G, we can basically determine what amino acid will come next in the sequence. And based on whatever your amino acids are, methionine, lysine, isoleucine, aspartate, phenylalanine, whatever they are, those are going to be your amino acids that those R groups determine how your protein is going to fold. Remember, our primary structure is your amino acid sequence. Secondary is those alpha helices and beta sheets that are determined by the R groups right next to each other. Tertiary is how they're going to fold overall and create disulfide bonds and stuff like that. And then quaternary is whenever one polypeptide works with another one to make a working enzyme, for example. Okay. So I'll remind you of amino acids. If these are amino acids, remember an amino acid. And we thought that we were done with chemistry. Please. Okay. So here we have an amino acid. We have NH2. Here's your NH2 at your N terminus. We have a central carbon that's attached to your R group. We have another carbon that has a double O and an OH. This is what an amino acid looks like. This here is an amine, and this here is a carboxylic acid. Amino acid. Okay. So what happens next is during that peptide bond formation, nope. This O comes in and binds with that N. And it's going to be another one here. It's the same exact structure. It's just, you get the idea. Okay, so one amino acid links to another amino acid. What's this bond here? That's the peptide bond. That's what's really happening in translation. Whenever we link methionine to lysine, it's because we're creating a peptide bond between them. Are we following? Hope so. So that's what's really happening. Those R groups just determine whether it's methionine, lysine, isoleucine, whatever. But all amino acids are the same except for the R group. And R group dictates what amino acid you are. So this is happening in the ribosome. The ribosome clamps down on the messenger RNA, and then the tRNA comes in and matches with the codon. Its anticodon matches the codon and says, here's the amino acid that goes here. Then the next one comes in and they form a peptide bond to each other. Once they've peptide bonded, the original tRNA can skedaddle off. And eventually we'll be left with a polypeptide that can be part of a functioning protein. Any questions about translation? Okay, I'm going to erase some of this stuff in the middle. All of our amino acid stuff. Whatever, we're making space. All right, we got some questions about translation next. We have barely made it through that on our questions. All right, codons is 3 bases on what molecule? The codon is on the I'd have just fast forwarded to this picture. I'm just realizing. All right, so beautiful. That's on the mRNA. So here's our mRNA. It's this red thing here, and we're matching up our tRNA anticodon to it. It carried with it and amino acid, and it'll link to the next guy, and then just keep building the chain and keep shoveling down. Again, there's a video on this on that concept map page that I highly recommend that you watch. It's better than my drawings, okay? All right, next. Question 9, operons. Don't you love it? Okay, in operons, repressor proteins bind to operators unless a molecule that changes the repressor shape is present. What type of operon does that describe? Do your best guess with this. We're going to go over all the operons next. All right, it's okay. There's two beautiful guys. I love you. All right. So there's two types of operons, and I'm going to go through first to start with operons in general, and then we're going to get to the two different types. Okay. So first of all, we have enough space on DNA in our double-stranded DNA. Remember when we're going through transcription, we're going to open up that DNA at that spot where our promoter is. And then our RNA polymerase will transcribe. Okay, so first things is the promoter. Then we have something called an operator. And then we have whatever our genes are, gene 1, gene 2, and gene 3. This is a rule bacteria follow. In our cells, eukaryotes, ours are not set up like this. In eukaryotes, we will have a promoter and other, regions, kind of like an operator, that control gene expression for every gene individually. But bacteria can lump them under one controller. Okay, so this is bacteria only. Come on, pen. Bacteria are the only ones that have operons. Okay, so promoter, operator, and the genes. So the promoter is still the same thing as it always was. This is where RNA polymerase binds. So in order to open up the DNA and make an RNA copy of the DNA, that's where the promoter binds, okay? So we'll make our promoter in green, or sorry, our RNA polymerase in green, it binds to the promoter. All right, so it would open up and make a copy of gene 1, gene 2, gene 3. Instead, we have this red guy here, which is our repressor. Now, because that red glob is sitting there, we can't get to gene 1, gene 2, gene 3. It's physically blocking us. So this is how all operons work, all of them. The differences come in how they start. So in an inducible operon, you're going to start off. When the repressor is bound and we can't make our RNA because RNA polymerase is stuck, then we are off. We are not expressing our genes, okay? So you're an inducible. So think of it this way. If you have to induce labor in a woman, was she in labor? No, you're turning labor on, right? So she started off and now we're going to induce her. She is inducible. So we can turn it on. One thing we can say is we can remove the repressor, but that's another thing. So you can turn on. Okay. In a repressible operon, the idea is that you start on. It's the opposite of inducible, quite literally. And you can turn it off. Okay, so let me draw them out for you. So I say draw them out. We're going to use our same example. An example of the inducible operon is our lac operon. It's the one that's given in the book for us, okay? The lactose operon, the lac operon, is an inducible operon. It starts out just like our drawing is drawn right here. It starts off, and we want to turn it on, okay? So the repressor is bound. What happens is lactose comes around, and it's going to start as our inducer. Inducer. It's going to induce by removing our repressor. So what happens is lactose binds to our repressor, The repressor, like many proteins when they bind to things, then changes shape. And it comes off. Chew off. And now our RNA polymerase can work. We're following? Okay. That's how that lactose one works. With the repressible operon, it starts on. So it starts just like this. An example, one of the examples that it gives in the book is the arginine operon. Arginine is an amino acid. So this is one sort of like our meatball and spaghetti example, okay? You have your book in your pantry, which is our DNA here, and your promoter is whatever that marking on that page is that shows you where meatball and spaghetti is, the tab that sticks out that you're going to open it to, okay? But if there is a lock on the book, the recipe book, that's our repressor, okay? So in this case, there's no lock on the book. What's happening is you keep making more and more copies of the recipe. Meatball and spaghetti, wrote it down. Meatball and spaghetti, wrote it down. Meatball and spaghetti, wrote it down. All right, everybody, get ready. You got your copies of meatball and spaghetti. We're all going to make meatballs and spaghetti for a big old party, okay? And that's what's going on. Well, let's just say now all of our guests at our party are leaving. They're all full. They don't need any more meatballs and spaghetti. If you keep making copies of the written recipe, nobody wants to eat the meatball and spaghetti, and nobody needs new copies of the recipe written down. Why are you keeping making copies of the recipe? So we will lock the book so that we can't keep making the recipe. And that's the idea there, okay? So what's happening is arginine is an amino acid. If we're not building protein that needs arginine, why are we making arginine? Or another example is factory. The factory is making summer dresses, and now we're in winter. Nobody's buying summer dresses. Why are we going to keep making summer dresses? Okay, so eventually what happens at the molecular side of things, you have the shop manager or the person that's watching over the cookbook or whatever starts noticing that you're building up excess arginine or spaghetti and meatballs or summer dresses, whatever you think, okay? So we have too much summer dresses, we have too much spaghetti and meatballs, we have too much arginine. It's not getting used up. So what happens is when it's not getting used up, it's available to bind with the repressor, basically signaling the repressor, the lock. The lock will then change shape. And we know locks actually literally work that way. You change their shape and they're either locked or unlocked. So here we're locking it. So they literally work exactly opposite of one another. All right, you guys tracking? Does that help at all with the operons? I hope it does. Focus more on inducible operon versus repressible. Don't focus so much on lac operon and the other names of the operons, okay? Focus more on inducible operon versus repressible operon, okay? Let me make sure I'm not missing anything. Good, all right. Questions about the operons? Okay. Last one's about mutations. Missense mutations. What are they? Let's keep going down. All right. So we have several different kinds of mutations. Let me draw some of them on here. Easy enough. Okay. Missense. It's just like this says. If you had your original, we'll do the original up here. If your original code was AAA and you coded for phenylalanine, codon was AAA and your amino acid that you coded for was phenylalanine. If you went and changed one of those nucleotides, and now you're GAA, and now you're coding for isoleucine, right? Change one nucleotide, you change the amino acid, that's a missense, okay? No, come on, behave. Silent mutation. If you went from AAA to AAU, I shouldn't put T, so forgive me for that. AAU. And now you still code for phenylalanine. That's a silent mutation. We changed the nucleotide, but we didn't change the amino acid. That's a silent mutation. Okay. A nonsense mutation. Nonsense codons are codons that do not code for an amino acid, and they therefore cause the translation process to stop. So we call them stop codons, okay? So if you went from AAA to UAA, I don't know, and then that was stop. That's a nonsense, okay? If your original was AAA and you switched to GAA and then turned back into AAA, that's a back mutation. For example, going back to the original, okay? If your original was, let's say, even longer, I have to, just trust me, okay? AAA, CCC, GGG, and this was your whole sequence in your DNA, in your mRNA result, okay? And now we have changed it, so we have deleted one of these A's. And now you're AA, you don't skip and keep on with CCC. You move it over one. Now you got CCG, GG, whatever the next one is. Now we've changed this one, and this one. By removing one and shifting them over, this is a frame shift mutation. You can see how devastating this could be, right? Obviously, stop mutations, nonsense mutations, and frame shift mutations, the most devastating of all of these, okay? Any questions about the mutations? This gets us into talking about those single nucleotide polymorphisms. So that was in chapter 8, right? And we talked a little bit about, it might have been in this, I think it was in chapter 8 when we started talking about gene therapy and started getting into single nucleotide polymorphisms, which are SNPs. It's a fancy word. This is to say that in some of our genes, one single nucleotide can lead to many versions of a gene. So whether this is AAA, AAU, AAG, AAC, might code for different amino acids or even a stop, and those different versions of the genes are different products that can result. And so this single nucleotide polymorphism, polymorph just means many versions. So many versions of genes from one nucleotide changing that can be deadly, or it can just lead to a different version of the trait. Could lead to a different eye color, could lead to being taller, but it also could mean you can't clot your blood or that you clot it too much, which was the example in the book. That somebody had a mutation of one nucleotide that led to basically excess thrombosis, so excess clotting. And if you have too much clotting in your blood, that can lead to strokes, high risk for strokes. Just one nucleotide change. There are some nucleotide changes that can be deadly and you wouldn't even grow in the womb. Nobody would even know you were a baby. It would not be viable. There are some nucleotide changes that just one nucleotide of a change can lead to, a non-viable fetus. So that's a single nucleotide polymorphism, and that was talked about chapter 8, but I feel like it relates to these here, so I wanted to bring it up now. Okay, any questions about that? All right. But I don't remember which chapter. It might have been chapter 8, it might have been chapter 6. Doesn't matter, but you got to learn about it now. All right, so good. Here's our podium for chapter 6, and we will do our exercise for chapter 6.
Engineering, and they just put out a report that one of the regional something like blood tests to test for concussions, and they use like genetic biomarkers and stuff.
Wow, yeah. So that's another thing, right? If you've had an injury, it might trigger certain genes to be expressed because we need to heal certain things. And so if you're expressing those genes, it could be an indicator that you had a brain injury or something like that. Yes. And that's how all of our body works. We just forget, or we don't really relate it in the big picture fashion, right? The only reason I'm talking right now is because some signals were being sent to my cells. Literally, it's all chemistry. I hate it, but you gotta love it too, right? That's biochemistry, man. The only reason I'm even turning my head right now is chemistry. Alrighty, cool. So let me pull up chapter 8 before I get too far. We'll have it in the background and we'll come back to it in just a second. But before we get too far with that, I want to go over our activity. So for chapter 6 activity, we have HIV-1 reverse transcription. And I try to pick where I can recent up-to-date articles, but this one is one from basically like looking at an overview of the history of HIV. So this was written in 2012, but at the same time, it is basically an overview of our understanding of one of the most important enzymes that we've ever discovered in all of science, and is made by the HIV virus, reverse transcriptase. Okay, if you want to read through this page, again, I only give you one page of these. If you want to read through it, you can. I'm not going to read the whole thing. But 40 years from 2012, okay, we discovered reverse transcriptase. And it was basically announced by Howard Temin and David Baltimore from the university that I actually studied with. So pretty cool stuff there at UPenn. So anyways, David Baltimore and all this group here. Cool that they did all this stuff and it won them Nobel Prizes and all of that. And here is why, because this is really fascinating stuff, all right? HIV is a virus. It has a genome. Well, we know all cells ever have DNA as their genome, but viruses aren't cells. They're acellular pathogens, right? And as it turns out, HIV has a genome that is a positive sense, single-stranded RNA. That's in the article. Don't expect you to know that off the top of your head, okay? Not yet. You do need to know it for Unit 4. That's why I have this kind of stuff that you need to get to know for Unit 4. It's a lot of recall stuff, but anyways, it's a positive sense single-stranded RNA, which basically makes it equivalent to mRNA. It means that a ribosome can translate it. So this brings us back to that concept of the different types of genomes you can have. If you are a negative sense single-strand RNA, we would have to make a copy of you first. So what I mean is if you're negative sense single-strand RNA, if you were AAU, whatever, and then we would need to turn you into UUA first, and then the ribosome can read you. We need to make your complement, and then the ribosome can read you. That's what negative sense would mean. If you're positive sense, the ribosome can just read you. All right. We're positive sense single-stranded RNA, which is equivalent to mRNA. This means that ribosomes can translate them. That means that we don't need to make copies of our genome. A ribosome can just see, hey, you look like mRNA, bind onto it and start matching codons with amino acids and start building proteins right away. That's what it is to be positive sense single-stranded RNA. But it gets so much more beautiful, okay? HIV produces something called reverse transcriptase. So if we know that DNA to DNA is replication, and DNA to RNA is transcription, and RNA to protein is translation. Reverse transcription means that we are going from RNA to DNA. And the enzyme that drives that is the reverse transcriptase. Okay? So HIV takes this little old RNA genome and reverse transcribes it back to DNA. Now that's wild. And that went against everything that we knew about how the cells work. So this was a massive discovery that this could even happen. Any virus that can do this, we call a retrovirus. Starting to sound a little bit familiar? Okay. That's a retrovirus. So what happens is we'll turn this into DNA and then into your little host. So I'm going to draw a little box out here. This will be everything that relates to question 2, okay? Into your little cell. This is your cell, and this is your nucleus, And this is your DNA in here, okay? This will take that HIV DNA and insert it into that DNA. The DNA copy of HIV. So if we insert the DNA copy of HIV, it's not HIV, it's not the virus that's in your DNA, it's the DNA copy of the virus's genome. You guys remember the term for that? It was on the test and a lot of people missed it. It was provirus. Once it's in here, this HIV copy is a provirus. You love it, okay? So it's latent, it becomes latent. This is lysogeny. It is the same idea, okay? We can use that to make RNA versions of that HIV and build new viruses. Also, when the cell replicates, if the cell replicates, it'll cause the replication of the provirus. So each time that that cell replicates, we're just making copies of the provirus. You guys following? Okay. Anybody know what types of cells HIV infects? Oh, I forgot I can't scroll with this stuff on here. Anybody know what types of cells HIV infects?
Isn't it T cells?
T cells. So they are specifically CD4 positive T cells. And we don't need to know right now all the details of what T cells are which. Don't worry, that's coming in Unit 3. Don't you love it? Okay. But it specifically infects our CD4 positive T cells. CD4 positive, that part of it is important because not only is that the type of cell they infect, but that's the lock that they unlock with their spike protein, is the CD4 molecule. The CD4 molecule is used by your T cells to basically tell your body what we're interacting with a bad guy. Okay, that's really watered down version, and we'll talk about really what all is going on molecularly in Unit 3. We're not there yet, okay? But it uses that as a way to get into the cell. And it is a marker found on our T cells, one type of our T cells, called our T helper cells. But don't let the fact that they're helpers belittle their purpose in your body. Literally, your entire immune response, whether you make antibody or whether you kill infected cells in your body, is governed entirely by your helper T cells. So if you have HIV, you're killing the captains that govern your immune response, and you cannot have an immune response to anything new. That's why people with HIV develop AIDS, acquired immunodeficiency syndrome, because we're killing all of their immune cell captains with HIV. Okay, so it embeds itself into those immune cells' genome. It's not going to infect any other cell in your body, just those T cells. So those T cells, their DNA gets changed, but not any other thing. In order to insert the DNA into that cell, we have to use another enzyme. It uses another enzyme called integrase. Now you can see why, this is a S. Now you can see why I get so frustrated when somebody says mRNA from a COVID vaccine is going to get inserted into your DNA. When HIV has to have two separate enzymes that it specifically codes for just to make that happen, and it only gets into your T cells. And it only affects your T cells. You're not going to have HIV DNA in like your eyes or your liver or anywhere else, only in those T cells. This is a very gated, unique process associated with retroviruses in general. HIV is not the only retrovirus. There are several. In fact, there are other viruses that aren't even RNA viruses that can also make reverse transcriptase, which is even wilder to think about. This includes hepatitis B. Okay. So hepatitis B also makes reverse transcriptase. It's a DNA virus, and it takes its DNA, turns it into mRNA for building things, and then takes that mRNA and turns it back into DNA that it inserts into your body. And that's how we get cancer from hepatitis B. It's also why we can't cure hepatitis B. Once you have hepatitis B, you have hepatitis B for life. There's no cure for hep B. There is for hep C. It's different. Okay. But we do have a vaccine for hep B. No cure, but there is a vaccine. Okay. So anyway, so this all relates to that. But HIV was the first one that we discovered that did this, and it was groundbreaking. The fact that it could go in reverse. What do you mean? So, good stuff. Nope. Clear this. Next question on here. The study mentions reverse transcriptase as the most common target of antivirals to treat and prevent HIV infection. We actually use multiple drugs that target reverse transcriptase and even drugs that target other aspects of HIV that are unique to HIV, like integrase. All right. And it's so effective. But if we could prevent you from getting the HIV, that's what the goal would be, right? If you can't reverse transcribe, you could get HIV virus into your body, but you can't turn it into DNA and incorporate fully. We can protect you from having HIV. So that's what PrEP and STEP are. You probably heard about them in the TV. And any of those that are preventative HIV medications that you can take if you are going to be putting yourself in a lifestyle that puts you at risk for that. If you're having a partner that you know has HIV, we can put you on PrEP or STEP. And you can take that and it prevents you from getting HIV. It's actually very effective at that, okay? So why would that be an excellent target? Because obviously our cells don't have this enzyme. And so if we attack that, it shouldn't have any toxicity to our own cells. And this is true. I mean, you can take it as a preventative and you shouldn't have any negative side effects associated with it. Did you have a question?
No, it's crazy to me like how we discovered this only about 40 years ago, like the 80s, and now you can have a child being HIV positive and that child doesn't have to worry about
Yep.
it at all.
Yep. If you are a mother if you're HIV positive mother, you have HIV, but we put you on the medications, it's non-toxic to the baby, and the baby won't get HIV. As long as you're on those medications while you're carrying the baby, you can give birth to that baby, and as long as you're on those medications while you're breastfeeding, that baby has a 97 to 98% chance of not having HIV. That is wild. And that is beautiful. So that's something definitely to be aware of. And I think it's even getting more and more effective as time goes, as we develop more and more drugs and more awareness about this stuff too.
Is there like a point to where it would be effective?
If the virus mutates. And so we have seen instances of that. There are some strains with the original drugs that we originally targeted with this. The virus was able to mutate its reverse transcriptase. So it was like evading the drug basically. So that can happen, unfortunately. So far, the drugs that we have been pretty steadily reliable, as long as you have like more than one. You know what I mean? If you're using just one drug, then you're likely to have mutation. But if you're using multiple, it's unlikely that they can mutate to evade all of the drugs, if that makes sense. So that's usually why actually we use more than one drug with HIV treatment, is for that reason. Imagine how quickly our bacteria multiply and how quickly they can evade antibiotics. Well, think of if you were a virus and how quickly they multiply. I mean, it's drastically faster. And so they have much more opportunity for mutations. And they're not a whole cell that they have to keep alive. They can manage to have some cost associated with having high mutation rates too. You know, if you have a mutation in your cells, for example, it could kill you. You're a multicellular organism. You probably need all those aspects that are for a virus. If they lose one virion to that mutation, what do they care? They've got a million more that they're building in the background, right? So they can handle mutations a lot more. It's not individually impactful. All right, reverse transcriptase can convert RNA into DNA. How might this be used in research and medical advancement? This does touch a little bit more into chapter 8, but also can be giving us this idea of thinking about now we can study viruses that have RNA genomes by converting them into DNA and manipulating them much more easily. So one thing that it leaves out of this is that working with RNA is very unstable. Just like in our cells, it's not meant to last in our cells, it's temporary. And that is the true, just overall, generally speaking, physics-wise, chemistry-wise, RNA is very unstable. And so this allows us to work to convert RNA back into DNA, and DNA is much more stable and easier to work with and manipulate. And so we can study things a lot easier that way. Another thing you could think of here, if we can use reverse transcriptase and integrase, then what we might be able to do is engineer an HIV virus that instead of coding for HIV crap, have it code for a working copy of a gene and put it into somebody that has a mutated copy of a gene where they couldn't clot things properly, where they had sickle cell disease, where they had some sort of a genetic mutation, and now put a working copy of that gene in their body by inserting it into their body using HIV. That, my friends, is how gene therapy works, quite literally. And so it doesn't code for HIV or its parts anymore. it codes for those genes that we want it to instead. So you're not at risk for HIV. You don't have HIV when we do that because it's not HIV anymore. We cut out all the HIV parts and just put in these good genes. And so we'll talk about that in our next activity for chapter 8. Okay. So we're already at 6:20. So we're going to go through at least some of the questions for chapter 8 really quick. All right, any questions about the HIV stuff? Yes.
So with the gene therapy stuff, part of the, like, modified virus is able to replicate anymore, or do they have to synthesize them before?
Right. So they will basically make those viruses. Like they can culture up a virus that can replicate itself, but it won't be able to make actual HIV version. You know what I mean? It won't revert back to HIV. It'll be a virus that has just the good stuff instead. If that makes sense. Yes. It's really watered down version of it, but yes, that's the gist of it. Yeah. And I think it's just super cool. Try and tell me it's not. All right. So we will, I think we're ready. Okay, make sure that you are signed in and we're going to get started with chapter 8. I think that's everybody. 1, 2, 3, 4, 5, 6. Jillian just went to the bathroom or something. Good. All right, first question for chapter 8. So genetic engineering stuff. What type of biological macromolecule are restriction endonucleases? I'll point out the ACE. We'll talk more about these types of macromolecules in chapter 10. But any of the ACEs are enzymes, and enzymes are functional proteins, right? They're catalysts, but they're protein catalysts. Okay? So it's okay if you didn't get it, but they are proteins. Next. Another question about restriction endonucleases, and then I'll tell you a little bit about them. Restriction endonucleases are what? If you guys know, then you know, but my hair has reached the end of the line with passing with dry shampoo. So we got to the point where I think I need to, you know what I mean? That's why I keep touching it, because it just is like almost like it's just got too much dry shampoo in it. All right. So restriction endonucleases are molecular scissors. I know that the picture kind of showed it, but the idea is that if you have a sequence that looks like CAATTG, okay, in your DNA, then the opposite side of your DNA, if this is your 5', this is your 3', right? This is your 3'. You would have G, T, T, A, C. Good little refresher on our genetic code there. But notice something. If you read this CAATTG in the 5' to 3' direction, now read this one in the 5' to 3' direction. It's the same. We see that? All right. This is what we would call a DNA palindrome. It's a sequence that reads the same forward and backwards on the opposite strand. All right. Another thing you would notice is that if you folded this over, if you take just one of these strands, just one strand, and you fold this over, those would match to each other too. So this type of sequence is where restriction endonucleases cut. It's a molecular palindrome. And how they cut will be like this. And there's another picture in the next question that depicts this a little bit better. Okay. But they'll cut like that, and they create a sticky end. Right. So you'll have this overhang of AATT, AATT, and you'll have another overhang of TTAA on the other side, and then come apart. And if you wanted to rejoin them again, you'd need to rejoin them using DNA ligase. DNA ligase is always what rejoins our DNA, okay? But otherwise, after you cut with that enzyme, you have this little sticky end. Now, one enzyme, I don't know which one, let's just say it's E. coli enzyme number 1. cuts at this specific sequence. Another enzyme that maybe comes from Staphylococcus aureus cuts at a different palindromic sequences. So each restriction enzyme cuts at one very specific and defined site, palindromic site. So restriction endonucleases is any group that falls into this. Sometimes they create sticky ends like this. There are some that create blunt ends, so they just cut right down the middle. But you can see how this could be useful. Imagine that I have the gene for insulin. Now all I have to do is add TTAA at one end, and on the other end, add, AATT or whatever, and I can stick this in, into the bacteria. And now I can make the bacteria express insulin. And again, that's an oversimplified version of that explanation, but that is quite literally how we made bacteria produce insulin. It's where we get insulin from. So we took a plasmid, more specifically, a little extra bit of DNA, cut it with an enzyme, and stuck human version of insulin in there. Now, if you read about chapter 6, you learned about introns and exons also. So eukaryotic DNA has introns and exons. So insulin has introns and exons. Bacteria don't know what to do with an intron or an exon. So what those are introns are extra crap in the DNA. We don't need them, and we'll cut them out of our mRNA. Bacteria can't do that. So we have to take mRNA of insulin that's already been processed. It's already cut out all of the introns and joined together the exons, usable mRNA from human cells, and turn it into DNA. And now we can take that spliced, already processed DNA and put that into the bacteria. You starting to see where reverse transcriptase became useful suddenly to us? Yes. So the only reason that bacteria nowadays can make insulin for us, makes insulin affordable, and makes it so diabetics can live, is because reverse transcriptase from HIV, a horrible disease that is incurable, that if you don't take on your medications regularly, is 100% fatal if it's not treated. We take that disease, took something from it, and now we can save lives with that technology. Give and take, that's what life is, right? It's a blessing and a curse. Okay, so this is the idea. So this is where it's useful, and this is where we learn about this in chapter 8, like how to use this technology to improve the livelihoods of people with knowledge of genetic information. Okay, so that's restriction endonucleases. They're also pretty unique in that they only cut at these specific sequences, but you'll just randomly have these sequences in your DNA just because of how your genetic makeup is. And if I use this specific enzyme and take all of my DNA and cut up my DNA, okay, I do that to your DNA too. Your DNA will have different cut points than my DNA will. And so when I run them out on electrophoresis, this might be Lauren, this is me, in case you weren't paying attention, that's my first name, okay? And this might be, I don't know, Duncan. And when I cut mine and run it through the gel, the big fragments, they don't travel as far, they get stuck in the gel. The little ones will travel farther. This might be my profile, but Duncan's might look like this. Who knows? Okay, just based on random sequences in our DNA. So now suddenly, just by cutting up somebody's DNA and running it on one of these gels and separating it based on size, okay, this basically can allow us to look at who done it or who was the victim or even things like that, right? So let's say you guys walk in, let's say, I don't know, Jessica walks in, okay, and sees me and Duncan standing here and everybody else in the classroom is dead. You might be like, I'm sitting here saying, no, Duncan killed them. And Duncan's like, no, Lauren killed them. She was sick of their arguing and not doing well on the test, okay, that she did. So now, Jessica, I mean, what do we do? We would say like, well, let's try and compare their blood on gel electrophoresis. Let's cut it up with 1 or 2 enzymes and see what their patterns are. And we can test blood splatter around the room. They both got scratches on them. Whose blood matches the blood of the killer or whatever, right? You get what I'm saying? So now we can try to determine what's going on. Hopefully one of you guys scratches one of us on the way down so we can look under your fingernails and find DNA evidence, okay? Just saying. All right. Always. This is like one of those more serious moments where I would say that if you are ever being attacked by somebody, even if you can't save yourself, you can save somebody else in the future if you take some of their DNA with you. Scratch them, bite them, whatever you need to do. Okay. All right, so easy enough. All right, so let's move on to the next question. It's going to have to do with our restriction enzymes. Restriction enzymes can cut anywhere in the DNA sequence. Is that true or false? I hope that you see what I've written up here. So we can agree. Can they cut anywhere in the DNA sequence or only at specific special spots? Yeah, so this is false. They can only cut at specific palindrome sequences. So that's our restriction enzymes. That's it. Kind of keep track. All right. Next. True or false? CRISPR-Cas9. This is not a restriction enzyme, okay? CRISPR-Cas9 is a protective system in bacteria that can be used in genetic engineering to cut just about any sequence. Is that true or false? We talked about restriction endonucleases, also known as restriction enzymes. But what about CRISPR-Cas9? I know y'all heard CRISPR. I don't feel like we can get away from it. So CRISPR is an enzyme, and it's from a gene sequence that works in conjunction with this gene sequence Cas9. Okay, so CRISPR-Cas9 is a system that we basically found in bacteria, just like restriction enzymes. So those were enzymes made by bacteria also that cut at specific palindrome sequences that were protective. They protected against foreign DNA and helped them, like, we don't want you in here, cut it up, get rid of it. CRISPR-Cas9 does a similar thing. But what it'll do is the cell can basically say, this sequence, cut out anytime you see this sequence. And then CRISPR says, yes, that sequence, takes that sequence, goes and compares it throughout the DNA. And anytime that it finds it, cuts. So it can potentially, if we give it the signal to find this sequence, cut anywhere in the DNA that we want it to. But you have to tell it what sequence you want it to look at, right? And we can cut anywhere in the DNA. And potentially can use it to cut out parts of your DNA and insert working copies as well. Potentially. Now, in animal systems, we have not been successful with this. Not long-lasting effects of this. But this is the wave of the future. This might allow us to cut HIV out of your T cells. This could cure HIV. It could cure other diseases as well. But imagine now, we'll get into it later. I don't want to get too far into it. Anybody in here seen the movie Gattaca? No. The first time that I've had class that nobody has seen it. Okay, no worries. We'll talk about it in a second. Or at least the premise of it. I won't go over a whole movie with you. No worries. Okay, PCR is a technique. What do we use PCR for? What does it do? So I'll help you out here. Vivo is inside of the body. This would be happening. Vitro means it's happening in a test tube outside of the body. Help you with that a little bit. We'll probably need this picture. All right. So the answer is that it amplifies DNA in vitro. So whenever I take a swab of your nose, I put it into something and it runs the PCR. We're not testing it while it's in your body still. So that's the difference there. So if something's happening in vivo. An example of a test that is done in vivo is your tuberculosis skin test. We're doing a test in your body, like actually in your body, okay? Very rarely are tests done in vivo. When I draw your blood at a hospital and somebody wants to run a CBC, we send that blood up to the lab. That's an in vitro test. We're not testing it while it's in your body. It'll test it while it's outside of your body. So that's in vitro. So that's the difference there. We also have some testing methods, one of which was talked about in this chapter, and which we will visit again in chapter 18 in unit 3. And that is the nucleic acid hybridization. Nucleic acid hybridization, where we take a sequence of DNA and see if it'll match up within your patient's sample or whatever, or your bacteria or whatever, looking for DNA matching sequences, okay? Not using antibodies, which we'll learn about in unit 3, but using DNA to see if it'll match up. That's nucleic acid hybridization. And we can do that in situ. And what I mean is I can take a breast cancer biopsy from somebody's lump in a breast, and then take that breast cancer biopsy and then see if a probe for a breast cancer gene matches in situ. Situ is like situation. not really in vitro because it's still in living cells, but the cells aren't in the body anymore. So we call it in situ. Okay, so those are the differences between those terms. In situ is just the one in between. Okay. In vivo is in body, vivo is alive, vivo. Vitro, I just think of it as also known as ex vivo, outside of the body. Okay. All right, so PCR. All we're doing in PCR is making copies of DNA. And that should sound familiar because that's what we did in replication as well. So PCR is just mini replication. So we're going from one double-stranded DNA molecule in one cycle of PCR. You will now become two. All right. In a second cycle of PCR, this one becomes two and this one becomes two. In a third cycle of PCR, I mean, you're going to get this real quick, I'm sure. This one gets two, this one is two, this one is two, this one is two. So it doubles whatever you had in the previous cycle every time. So after one cycle, you go from 1 to 2. After 2 cycles, you went from 2 to 4. After 3 cycles, you went from 4 to 8. Then you went from 8 to 16. And you went from 16 to 32, and so on and so forth. Okay. What's actually happening in the cycle? You will have your DNA, double-stranded DNA molecule, and you're going to heat. You know this, delta. This is my sloppy delta symbol. Okay. You're going to add heat. This is going to separate your strand. This is a step known as denaturation. The first step that we're going to do, remember how polymerase doesn't add on to nothing. You're going to start with a primer. In order to do this, we got to lower the heat a little bit so the primer will stick. So this is a step that we call annealing. Then we will elongate, which works the exact same way as it does in replication. You're just going to add on nucleotides. So you had your old strand, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and then your new strand building off of it in one direction or the other. Whatever. I drew them the opposite, but you get the idea. Okay. 3 steps. First step, denaturation. Second step, anneal. Third step, elongate. Notice how you have to have primers. The primers have to match the sequence. So in order to elongate, your primers have to match the sequences. Primers are usually about 30 nucleotides long. I drew them 2 here, but they're usually about anywhere from 15 to 30 nucleotides. They need to match a specific sequence. So you'll have primers that will build off of a COVID gene. You have primers that build off of a flu gene. You'll have primers that build off of an RSV gene. You have primers that build off of Staphylococcus aureus gene or whatever. You'll have to have a primer for that specific thing. And if you're testing multiple things in one PCR, which is incredibly rare, but we do have one system that we do that regularly with, and that is with COVID, flu, and RSV. You can run that in one PCR machine. Bet that they are not doing that for you unless you have severe respiratory disease. Usually little babies we might do that with, okay? They're not doing that for you when you go up to the urgent care because this is going to cost money to run this test. Like hella money, okay? We're talking like probably a few grand just to run this PCR test if it's multiple like that. So instead, we would rather just run one PCR test and charge you $500 for it, $300 to $500. It's not cheap to run a PCR test. Okay. So anyways, you have to know what you're testing for, and it tests for a specific gene, and that gene is usually associated with a specific disease, for example. So you don't just go to the doctor and say, I have a respiratory infection. They're like, pop it in the PCR. It'll tell us what it is. No, the PCR just tells you whether it's positive or negative for COVID, and that's it. If you have COVID, the primer will bind, and you will elongate. If you don't have COVID, the primer won't bind and it won't elongate. And that is it. It is way simpler than you guys ever thought it was. Did it copy it or not? If it copied it, you have the thing. If it didn't copy it, you don't have the thing. Each A, T, C, or G in the solution that we have mixed all of this up in is fluorescently labeled a different color. So if it adds an A, it'll fluoresce blue. If it adds a T, it'll fluoresce green and whatever. So we can actually watch in real time whether we're adding an A, T, C, or G. And if you're adding the stuff, we can very quickly, within a matter of maybe 10 to 15 minutes, tell if we're adding nucleotides or not. But we let it go for an hour so that we can be sure that it's actually adding nucleotides and it's not just a false thing. Okay. So that's why PCR takes like an hour to do. We can do this to see if you have the gene for COVID in your patient sample. If you have just one COVID DNA in your patient sample, this will pick it up in an hour, easy. Now, no home test that you do is going to be able to test positive for one virus. None of your home tests are ever that sensitive, but PCR is just one virus and it'll pop positive on a PCR machine. And the likelihood of it being a false positive is very low. This is a very, very sensitive and very specific test. Okay. So it's good for that, but it only tests for that one thing that you're testing for. And like I said, there are some cases where we can run one for multiple things. I know what time it is, please. Okay. Any questions about that? Okay. I'm gonna leave it at that. They're all drawings. And just really briefly, I know what time it is, just trust me. We'll be done with this. Escape. I'm gonna let you see the little podium for a second. Yay, there's your podium. Okay. Really briefly, chapter 8. This talked about gene therapy, and it is using gene therapy basically to treat sickle cell disease. All right. So it talks about multiple different genetic diseases that could potentially be treated with gene therapy, but here is talking specifically about treating sickle cell disease and using basically this situation in treating sickle cell disease, which is found most commonly in African heritage populations. So Black populations, we would call it here in the United States. So if you have Black heritage, African-based heritage, and you have increased incidence of sickle cell disease. So this is a gene therapy situation where we're using basically HIV as a vector. We're taking their bone marrow out of their bones. We're taking like you would with a bone marrow transplant, taking some of their bone marrow out, infecting it with our engineered HIV virus. Now, instead of making HIV, it inserts genes that help with making proper red blood cells. Okay. And where do your red blood cells come from? Your bone marrow. That's where all of your stem cells are for your blood cells, all blood cells, red or white. Okay. So we'll take your bone marrow, infect it with our new HIV that has that correct gene in it, and then infuse it back into your body. And now suddenly your bone marrow can make correct red blood cells, and it works. Okay. So here it talks about that. This is now affecting cells that are already made in your body. And then anything that they multiply from then on will have these traits. But do your sex cells that you transmit to your kids come from your bone marrow? No, they come from your testes or your ovaries. And if you're a woman, then you made those eggs while you were in the womb. You ain't making new ones after that, right? So this is only affecting the body cells. So we call this somatic gene therapy. Somatic gene therapy affects existing body cells. Germline gene therapy affects the gametes and is heritable. Okay. All right. So here we're talking about sickle cell disease. Obviously, what are the benefits associated with this? Making your blood cells the way they're supposed to be. In sickle cell disease, instead of having red blood cells that look like kind of like this, their red blood cells look like this. They don't carry the oxygen correctly, and they cause damage to the spleen. They get caught in a lot of your capillaries, causing major pain, joint pain. It's an actual one of those diseases that causes such debilitating pain that is almost untreatable levels of pain. And so you can cure people of this with this gene therapy. Beautiful. It costs $1.5 million a person if you want this done. We can cure you for the rest of your life, but it costs you $1.5 million. Obviously not going to be FDA approved, because how do you do this much, research on a large group of people? How do you afford to do that in the clinical trials to get it FDA approved? that sort of stuff, okay? So that's an issue. And since this is a disease that exists largely in African countries, who's going to fund that? Right? So it also talks about successes and failures of several gene therapy trials. There's some successes of one person and then one person that has a bad experience with it. So we can already see this. Let me equate this to something that you're all aware of. How many people are you hearing from nowadays, increasingly, I am sure, that don't want to get vaccines because they're hearing about how dangerous vaccines are? So same thing here. If I were to tell you guys, hey, we're going to use a modified HIV virus, and I'm going to cure you of your sickle cell disease with that. How many of you guys, maybe previous or know somebody that would hear that and be like, I'm not getting HIV, forget it. It's the same thing as with our vaccines, right? Like, oh, that's going to change my DNA. Oh, that's going to do this, or that's going to do that, or I'm going to get autism or whatever. So it's the same kind of thing here, right? So that's what question 4 is getting at, aside from the cost issues, right? So then the last one, question 5, in what ways do legal, ethical, and social issues extend beyond affordability as well? So here, if we can just change somebody's genetic information, should we? Do we know what the long-term effects are going to be for them at the genetic level? Number one. Number one, here we're talking about this situation where it's a life-threatening disease that we can change. But now we've developed something where we can change somebody's genetic makeup. For now, maybe whenever you and your husband are ready to have kids, you go to your in vitro clinic and you pick, I want the baby to have blue eyes. I want them to be this tall. I want them to have a low incidence of getting fat. I want them to be an astronaut. Design them to be an astronaut, have all the traits that are desirable for an astronaut. And you can afford the $1.5 million to design your baby. But then the other people in the US can't. And now their babies have to be trash people because, and whatever. And that is literally what the movie Gattaca is about. If you haven't seen it, people start designing their babies. Ethan Hawke and Uma Thurman are in that movie. It's a great movie. It's old, to be clear. Like I say old, to me, like it's not old, but yeah, to you, it would be old. Yes. But basically, like there's one brother that was genetically designed and one brother that was born naturally. And the naturally born brother wants to be an astronaut, and he is sure he can prove that he can do it. So he finds a guy that is genetically astronaut material, and he starts saving up his skin cells and his hairs and starts leaving them around the place. But he makes sure that he cleans himself. So when they come testing to make sure, are you actually genetic material for astronaut? All the signs are there that he is, but he's using somebody else's genetic information. It sounds ridiculous, but it's worth seeing. And ends up that the guy has this heart defect. And to me, if you're going into space and now your team is relying on you being healthy to go into space and you're going up there with this heart defect, maybe you're being selfish, but also at the same time, bring up that idea of being able to select that your babies are blue-eyed, blonde-haired, tall, fit, all of these things. We call that eugenics. And there's obvious ethics involved in that. If you don't know where the idea of eugenics came from originally, it came from the Nazis. Yes, yes, yeah. So anyways, yeah. So let's head up to the lab. That's it. Try to keep lab as short as I can today, okay? Thank you guys. Beautiful.